From the top. Hello, my name is Matthew Lehrman. Welcome to my Survey of Biology final. The purpose of this presentation is to educate and build awareness of the biofuel industry and the process of biofuel production. This process is heavily reliant on the study of biology, as the main focus of the industry is the mass production of eukaryotic algae cells with high lipid content. The propagation of algae requires a solid understanding of biological processes like cellular respiration, replication, and cellular makeup. Further, the entire bioproduct industry is still relatively new, so much of the work being done currently is focused on developing efficient and economical processes to bring this exciting, high potential, and ecologically friendly resource to the market. As such, the many biology labs around the country conducting experiments to perfect this new technology are heavily reliant on the scientific process as it relates to biology, and a robust understanding of the study of biology. I will be outlining 4 main points in this presentation. These main points are as follows. What is biofuel? Biomass production, biomass to biofuel, and the economies of scale for the biofuel industry. The first point is to define biofuels. At the moment, biofuels make up just a small part of the larger bioproduct industry. When we talk about biofuels, we can be describing a number of different products that are all aimed at replacing conventional petroleum fossil fuels. All of the different biofuel products are derived from plant-based fats. This includes used cooking oil, often called biodiesel, sugar alcohol products from vegetables like corn and sugar beets, called ethanol, and lastly, my personal favorite and the subject of this presentation, biofuels from high lipid content algae cells. Now, algae is a promising candidate for the production of biofuels due to its high rate of replication, its abundant availability in a great many varieties, relative resilience to adverse conditions, and its eukaryotic form that allows for high lipid content. However, not all algae are suitable for biofuel production. Most algae species do not possess lipid content high enough to make biofuel production economically viable. Even among the most suitable species, there is left room for improvement in terms of the perfect biofuel producing algae. For this reason, in science labs across the country, scientists are using artificial selection to propagate desirable traits in the most suitable strains of algae in order to develop a production quality algae. Currently, chlorella is one of the favorites for the biofuel producing industry, although there are other algae species that possess desirable traits as well. The second point regarding biomass production. Algae is produced on a commercial scale in photobioreactors, or PBRs, from here on out. There are several different kinds of PBRs. I will be describing 3 of them for you here. All commercial scale PBRs use H2O as a biomass substrate, and most The simplest and cheapest PBR is called an open or raceway PBR. This kind of reactor is characterized by an open concrete pit that is oval-shaped and roughly 4 feet deep. This pit has a paddle wheel and an island in the center that forces water to flow around the raceway constantly. This agitation helps algae circulate through the solution and ensures that all the algae cells receive some direct sunlight exposure. Another reason for the paddle wheel is to help balance the gas levels in the substrate in order to maintain favorable conditions for algae to replicate quickly. The 2 main downsides of the raceway PBR design are the open air exposure and the depth of the track. The open air is a problem for several reasons, primarily exposure to the microbial contamination and risk of exposure to competing strains of algae that would use up resources while not providing the same level of high-quality lipid oils. This would ultimately lead to a reduction in the efficiency of production as well as lower quality product. The depth of the pit results in a condition where most of the algae in the pond is directly exposed. The depth of the pit results in a condition where most of the algae in the pond is not directly exposed to sunlight, consequently reducing its uptake of energy through photosynthesis and greatly reducing the potential biomass production efficiency. This phenomenon is known as self-shading. The raceway design has been tested for commercial scale production on several occasions and has proven to be a cost-effective way to grow and harvest algae biomass. However, due to the risk of contamination and the inefficient biomass producing qualities of the raceway reactor design, it is generally not considered a viable economic solution for commercial algae biofuel production in the long term. Another type of PBR is the vertical tubular type. This type of photobioreactor is characterized by many distinctive clear vertical tubes or cells that are filled with green biomass substrate. The vertical tube PBR alleviates the primary negative attributes of the raceway photobioreactor, although the vertical type PBR still has two primary drawbacks. First, the prior mentioned characteristic vertical tubes are arranged in a cubical form. This prevents all but the sun-facing perimeter tubes from receiving full direct sunlight. The vast majority of tubes are almost completely shaded from the direct sunlight exposure. This is not to say the shaded vertical tubes are not capable of facilitating cellular respiration, but the shaded tubes and the majority of the substrate is operating at a reduced efficiency in terms of biomass production. Second, vertical tube type photobioreactors are very costly to build. The high cost of production makes it difficult to recoup initial and operating costs, resulting in higher production costs overall. The vertical PBR is more efficient than the raceway type due to its higher surface area of exposed substrate per acre and its closed system, but there is one more photobioreactor to discuss. One of the most promising types of PBR is known as the Fibonacci type. This photobioreactor is characterized by a single clear plastic tube that is coiled around a conical-shaped frame. In this configuration, the substrate surface area is maximized while minimizing the self-shading effect. CO2 is added at the bottom of the coil to facilitate proper cellular respiration. At the top of the coil, the substrate enters an off-gassing and harvesting tank. This tank allows oxygen produced in cellular respiration to escape the system, as well as providing means for the biomass extraction when algae concentrations reach target levels. The Fibonacci PBR shows great promise for use in biofuel production, as it has been shown to produce comparable biomass quantities as other tubular type PBRs, but at a lower cost. The Fibonacci type reactor is also easier to manage with an increase in fine control over substrate temperature, pH, nutrients, and biomass extraction. Now we'll talk about biomass to biofuel. Let's talk about how we get biomass to biofuel. When biomass concentrations reach target levels in the PBR, a harvesting operation is needed. There are many methods of harvesting biomass from the substrate. The National Renewable Energies Laboratory outlines a process of using flocculation to pull the solids out of solution and utilizes dissolved air flotation to reach higher concentration. Although due to the harvested biomass then undergoes centrifugation. The harvested biomass then undergoes centrifugation, which increases concentration further. Although due to the National Renewable Energies Laboratory, the National Renewable Energy Laboratory outlines a process of using flocculation to pull the solids out of solution and utilizes dissolved air flotation to reach higher concentrations. The harvested biomass then undergoes centrifugation, which increases concentration further. Although, due to the significant cost and energy consumption during the centrifugation process, it is not economically feasible to incorporate this process in biofuel production at scale. Once the algae biomass is sufficiently concentrated, the lipid extraction process begins. To be able to extract the lipid oils inside the cell, sonication, or the use of sound waves to agitate and disrupt particles is effective in breaking down the cell walls. Once the cell walls are breached, solvents are introduced to isolate lipid oils from other biomass products. When the lipid extraction process is complete, the solvents are recycled and used again. The byproducts of this process are separated at this point to be used in different bioindustry sectors. For instance, the algae biomass less the lipid oil oils can be sold to biocosmetic companies for use in algae-based cosmetics. Biomass with high fermentable sugar content can be converted into fuel ethanol. The byproducts of the biofuel production process are known as co-products, and they are essential to lowering the biofuel production cost. At present, the favorite algae strains for biofuel production have been shown to be capable of producing lipid oil concentrations at up to 70% of cell dry weight. On the economies of scale, it's important to understand that biofuels are still relatively new. As much as we understand about the potential for biofuels, actually making this technology economically competitive is an entirely different matter. Current estimates, based on the data provided by several pilot facilities over the last 20 years, puts biofuel production costs around $40 per gallon. It is estimated that an industrial scale facility capable of producing 100 million gallons of fuel per year. It is also estimated that an industrial scale facility capable of producing 10 million gallons of fuel per year and using current technology could achieve biofuel prices between $10 and $20 per gallon. While this is still not competitive in today's market, it does illustrate the power of the economies of scale, the future potential of biofuels on the market. While this is still not competitive in today's market, this illustrates the power of the economies of scale and the future potential for biofuels on the market. With advancements in current capabilities such as balancing lipid content and growth rate, and finding and improving co-product scenarios, the potential for cost reductions is significant. In maintaining the growth condition, in manipulating the growth conditions inside the reactor, we see another example of the economies of scale. Conditions that create stress on the algae cells in the reactor can affect lipid content and composition, specifically the ratio of lipids, In In manipulating the growing conditions inside the reactor, we see another example of the economies of scale. Conditions that create stress on the algae cells in the reactor can affect lipid content and composition. Specifically, the ratio of neutral lipids, those used for long-term energy stores. In manipulating the growing conditions inside the reactor, skip this part, fuck that. In conclusion, the biofuel industry is exciting and shows much promise, although there is still a long way to go before this technology is ready for the consumer market. Investments in further research is needed before we can develop a process that will make biofuel price competitive with petroleum. Massive infrastructure investments are needed in photobioreactor and biorefineries in order to increase production capacity to meet demand. Also, developing co-product supply chains will help push biofuels into the ultra-competitive monopoly where petroleum products reign. The effects of balancing inputs during this production. The effects of balancing inputs during the production process to increase the concentration of nutrients. Gotta read it. In manipulating the growing conditions inside the reactor, we see another example of the economies of scale. Conditions that create stress on the algae cells in the reactor can affect lipid content and composition, specifically the ratio of natural lipids to polar lipids. For example, supplementing CO2 can lead to an increase in cell growth in the short term, but prolonged supplementation results in a decrease in the pH level, subsequently negatively affecting the growth rate. Another form of manipulating the growing conditions is irradiance. Studies show that with increased light exposure, there is an increase in cell growth rate and a small decrease in the neutral lipid production. However, the overall increase in growth rate results in an overall higher neutral lipid content per liter. Lastly, nitrogen limitation can cause algae to produce higher lipid concentrations in individual cells. However, due to the lack of nitrogen limitation can cause the algae to produce higher lipid concentrations in individual cells. However, due to the lack of nitrogen, long term, there is a reduction in overall cell count and consequently a reduction in marginal lipid quality. However, due to a lack of nitrogen, long term, there is a reduction in overall cell count and consequently a reduction in marginal lipid However, due to the lack of nitrogen long term, there is a reduction in overall cell count and consequently a reduction in marginal lipid quantity per liter. Nitrogen limitation appears to favor neutral lipid over polar lipid as well. In conclusion, the biofuel industry is exciting and shows much promise, although there is still a long way to go before this technology is ready for the consumer market. Investments in further research is needed before we can develop a process that will make biofuel prices competitive with petroleum. Mass infrastructure investments are needed in PBRs and biorefineries in order to increase production capacities to meet demand. Also, developing co-product supply chains will help push biofuels into the ultra-competitive monopoly where petroleum products reign. The effects of balancing inputs during production process to increase the concentration of neutral lipids is just one example of the advancements in the biofuel industry that are made possible by the study of biology. This is one of the many fascinating topics on the subject of biology that will inspire curiosity and drive the advancement of human understanding for generations. Thank you.